is work. Fine. So I'm here with a new topic. Okay, I think people will take a little bit of more time to come in. Okay, Mariam is here. Hi, Mariam. Welcome to the class. Okay, well, there is no response as of now. So I will have to wait for a few seconds and then we'll get moving with the class. Okay, hi, Mariam. So, how are things at your end? Okay, I think people may take more time to log in and we cannot wait till eternity, right? So let's get moving and let us talk about the topic that. Hi, Elena. Hi, Tasil. Finally, both of you came in. I was just waiting for you guys to come in. That's awesome. So, hi, Nivasni. Welcome to the class. Oh, today that's beautiful. We are more people than usual, at least. So I'm happy. And we have, okay, Christina also joining us. That is awesome. Hi, Christina. Welcome. Okay. Hello, Alina. Okay. So, guys, I guess everyone is ready and we shall begin now. And we are going to talk about our plants. Fine. So, till now, we have been discussing about control and coordination mechanisms and animals. And there we had talked of the nervous system of animals, right, which was playing a major role in the quick delivery of messages to and fro across the body and thus helping in controlling the functions of our various body parts. Fine. So we did talk about nervous system quite in detail and then we did talk about a system of control and coordination in organisms, and that is the endocrine system. So, in case of plants, I am in Guruvayar for my Arangatram. Okay, I don't know what is that, Nivasni. I have no clue at all. Anyways, whatever it is, take care and all the best. Fine. So let's keep moving. I think it's some kind of a festival or some kind of a ceremony which is going to be there. Okay, classical dance. Okay, all the best. Okay. So let us move forward and let us talk about how signals are perceived by the plant. So in my plants, the stimuli is perceived by the sensory cell and okay, awesome. By the sensory cells of the plant, the sensory cell will elicit or will produce some chemical and this chemical will basically activate some other cells and these cells will respond accordingly. Fine. So this is how the stimulus is able to bring a response in case of plants. Also, one more thing, a plant has got various kind of cells in various parts of its body and at different points of time, depending on the amount of water availability, the amount of moisture in the atmosphere and other conditions around it, some of these cells will start producing their secretions. Fine. So these secretory cells, I can say, which, depending on the physiological condition of the plant and the environment around, will start producing some chemicals which we often term as plant hormones. And these plant hormones are also called as phytohormones. And these phytohormones are released by these secretory cells, right? And these secretory cells are not a part of an endocrine gland. So in case of plant, we don't have the endocrine system at all. Fine. All we have is a group of secretory cells which will release a plant hormone or phytohormone in trace amounts or in very small amounts just outside of it. So the secretory cell or cells will secrete a plant hormone just outside of them into the extracellular space. And from the extracellular space, the hormone will move through the extracellular space to its desired tissue. And there, the effect of this chemical will be there. So, response will be there in the tissue due to the effect of this plant hormone or phytohormone. Fine. So, plant hormones or phytohormones are trace chemicals, not trace cells in trace amounts. These chemicals are released, fine, from the secretory cells in the immediate extracellular space, and these chemicals will keep on going in the extracellular space. They will travel and will move to their target tissue, and the target tissue will respond to the presence of this plant hormone or phytohormone. Fine. So generally in case of plants, the site of secretion and the target tissue are not far off. Why? Because here you don't have the blood vascular system to carry the plant hormones from one part of the plant body to another. So most of the hormones are released at the site of the action itself. So there are secretory cells around the place where action has to occur. Fine, they will secrete the plant hormone. The plant hormone will seep into the nearby extracellular space and will affect the tissue cells of the nearby extracellular space, and they will respond to it. So that is how the plant hormones work, and they are also released in trace amount or in trace quantities. Okay, so that is it, and let us talk more about it. So how do plant respond to stimuli without a nervous system? I think we have talked about it. So plants have hormones also called as phytohormones. These are chemicals produced in plants for triggering plant growth, or I can say for not triggering plant growth, for controlling and maintaining. Hello, Brian. Various physiological activities. Physiological means the daily activities required for living, like preparing of food by photosynthesis, respiration, fine, conduction of water, taking up of water from the ground or from the soil. So all these activities, the plant growth, the dormancy of plant, the senescence of the plant parts, the falling of leaves, fine, the formation of buds, the blooming of flowers. So all these various physiological activities are controlled and maintained with the help of many hormones. Alina, is it clear? So they are synthesized in minute quantities in one part of the plant body and are translocated or transferred to the other parts where they are required simply by seeping through the space between the cells. So that is how the plant hormone moves to their target tissue, right? Okay. Now, the two types of plant hormone or the types of plant hormones on the basis of function. So, there are two kinds of plant hormones. Some plant hormones are often referred to as growth promoters. And some are often referred to as growth inhibitors. Hi Priyanshi, welcome to the class. So some are also referred to as growth inhibitors. So now let us talk of growth promoters and growth inhibitors. And as their name suggests, they have different roles and different functions. So growth promoters include your auxins, gibberellins, and your cytokinins. And your growth inhibitors include your abscisic acid and ethylene. So we will talk about all of these things one by one in detail as we move forward. So I think the same story has been written here. So we have auxins, gibberellin, and cytokinins, which are the growth promoters. We have ethylene and abscisic acid, which are the growth inhibitors. Okay, now let us talk of auxins. So the word auxin means to grow, right? And auxin is a growth promoting molecule. So basically, auxins are a type of chemical which are secreted from the meristematic cells at the tip of the shoot and from the meristematic cells at the tip of the root. Auxins are secreted, fine, and these auxins will seep down, right, and they will cause rapid multiplication of the meristematic tissue beneath, right, and the meristematic tissue will multiply. So basically, just a moment. So basically, my auxin secreting cells are here, right, and the cells of my meristematic zone are here, below it, fine. And same is the case that is happening in my root tip. So I have my auxin secreting cells, few cells here, and rest all are again meristematic cells below. So the yellow cells are the meristematic ones. And these meristematic cells are activated when auxins come on to them from the secretory cells. So the secretory cells will produce auxins, and the auxins will impact the meristematic cells and it will tell them to undergo rapid growth and multiplication. As a result of this, the meristematic tissue will proliferate like anything and your stem will elongate. Right, so that is how the things work in case of meristematic tissue, so, or auxins. So auxins are released by the secretory cells above the meristematic tissue, and they will cause multiplication of the auxins. Sorry, they will cause multiplication of the meristematic cells, right? And uh, the shoot elongation and root elongation will occur. So, auxins are produced at the root and shoot apices and in young leaves also. And the functions of auxins, it induces apical dominance. So one function we have seen, it causes elongation of the axis of the shoot and of the root. Fine. Second is, basically it induces apical dominance. That means it will tell a plant to elongate its shoot in an upward direction, but it inhibits the sideways growth of the branches. So there is less sideways growth of the branches and more central growth of the branches in the presence of auxins. So if the auxins, so that means the auxins are telling only the apex to grow and the side branches will not grow much. Fine. So this phenomena is termed as apical dominance. And it is a result of auxins work. So it initiates rooting in stem cutting. So if you have got a stem cutting, let us say of rose, and you plant it in a pot. And after planting of the stem cutting of, yes, I know very well. So after planting the stem cutting of rose with a little bit of leaves here and there and a bit of a side branches, you start watering it for a few days, this stem cutting. So what will happen basically? New roots will come up. But the coming up of new roots can be increased quickly by the addition of auxins. So if you are also adding a little bit of auxins to your soil, right, or to the water that you are supplying to your soil, what will happen? It will quickly, the rose cutting, the stem cutting will quickly start bearing roots. So therefore, auxins also promote rooting in stem cutting, root formation in stem cuttings. It prevents immature fall of fruit and leaves. So it will prevent immature fall of fruit and leaves. So if your fruits and leaves are immature, and if your fruits are not completely ripe, your leaves are not old enough to be shed off, then they will not be shedded off due to the release of auxins. But it promotes shedding up of old, mature fruits and leaves. Fine. So that is a function of auxin. So these are some of the functions of auxins. And after this, let us talk about the work related to auxins. And this work related to auxins was carried out by Charles Darwin and his son. So Charles Darwin and his son published a paper in the year 1880, written as Darwin and Darwin. Fine. So basically what they did, they took a source of light coming from one end and supplied it to the seedlings, which are of some oats and some grasses. And we have seedling of some grasses. So, and what he did, the seedlings were exposed to a directional light, a light coming from one direction, and the seedlings were regularly watered and were regularly nourished. So the seedling starts growing in one direction towards the light as a result of exposure to light from one side, right? So that was there. And once these people, Darwin and Darwin, cut off the tip of the plant, they found that light could not influence the growth So the plant will grow upright and straight. Fine. So if you cut the tip of the stem here, the plant will not grow at all because the meristematic tissue is gone. So the plant will not grow here at all and will not be bent towards the light. Yes. And if you cover the tip of the plant with the black paper or the aluminium foil so that it cannot sense the light direction at all. In such a case also, the plant will not bend towards the position of the light but will grow taller naturally. Fine. So that is another thing which is happening here. So from all this thing, this Darwin concluded that exposure to light is making something secrete from the tip of the cells of the top to basically carry on the growth of the plant. Fine. So that is what they figured out through their experiment, and they were done by Darwin. Guys, are you all there? Come on. All right. So let's keep moving and let us talk of the next plant hormone, which is there in my list. The next one is gibberellin. And gibberellin was synthesized primarily in apical tissue and in young leaves. So gibberellin is also synthesized at the apex, at the shoot apex, right, by the cells at the shoot apex and even in the young leaves to promote their longevity, to promote their enlargement. And the functions of gibberellin include they can cause an increase in the length of the axis. So they will cause increase in the length of the axis. They will delay the process of senescence. That means cell death, right? So once a part of a plant has aged, it will prevent its desiccation or abscission from the main plant body, right? So that is the role of gibberellin. It induces bolting, that is intermediate elongation prior to flowering in beet, cabbage, etc. So what is bolting? Let us say I have two nodes here. Oh God. I have two nodes here and intercalary meristem on both the sides. So this is my node region. This is also my node region. This whole region between the two nodes is termed as internode. So this much is clear, right? Now what happens? We have the meristematic tissue here. This is called as intercalary meristem. And this intercalary meristem is going to quickly release some gibberellins. Is going to quickly release some gibberellins, and as a result of that, you will see something good happening. The distance between the nodes have increased. The distance between the nodes have increased. And this is the precise function of gibberellin. So gibberellin will elongate the internode. The internode has become longer due to the release of gibberellin from the intercalary meristem. Fine. Yes. And this happens just before flowering. So just when the plant is about to flower, just before it's flowering, there is an increase in its internode size. And this procedure of increase of the internode size just before flowering is called as bolting. This is termed as bolting. Okay, that is it. So it induces bolting in beet, cabbage, and some other plants, but not in all. Okay, so after gibberellin comes the cytokinin. The cytokinin is derived from the word cytokinesis. It is derived from the word. Oh, God, what a terrible day. Yes, cytokinesis. The word cyto refers to cell. The word kinesis refers to division. So therefore, this hormone, cytokinesis, will promote cell division, proliferation, and growth, right? It is found growing. So it promotes cytokinesis. They help in producing new leaves and chloroplast in leaves. So once this is released, it will induce formation of new leaves and chloroplast from the leaves. They promote lateral shoot growth and adventitious shoot formation. Hence, they overcome apical dominance. So basically, your auxins were telling the plant to grow vertically up and up. Fine. Whereas your cytokinins are telling the plant to grow dorsally more. It is telling the plant to go sideways, not dorsally, laterally more. So laterally you will start bearing more branches in your tree. And this is just the opposite of auxins. Fine. So also they will promote shoot formation in the adventitious shoot formation. So they will also introduce you to shoot formation. Let us see how. So if I have a dead piece of wood, right, which can give rise to another shoot, or a dead leaf which can give rise to another shoot, so apply some auxins to it. So application of auxins to the shoot will ensure that new shoots come up from the existing shoots or from the existing leaves. Fine. So new shoot is formed from stem cutting, leaf cutting, root cutting, and this is called as shoot formation, adventitious shoot formation, and hence they don't overcome apical dominance. They do overcome apical dominance. They help in delay of senescence of leaf. So they prevent delay in the senescence or falling up of leaves. So senescence is a growing up of a cell or growing old of a cell where it loses the ability to divide and it generally starts aging and dying on its own natural death. So that is senescence. Okay, what is senescence? Kindly repeat it, man. Let's make it quick. Looks like everyone is dull. Okay, anyways, let's move ahead. Okay. Now, effect of oxygen cytokinin ratio in cell culture. So what do I mean by this? Let us say I have a vessel and this vessel contains a semi-solid liquid medium. And this semi-solid liquid medium is very rich in nutrients. So this semi-solid liquid medium is generally agar, right? And it is very rich in nutrients. Nutrients has been added to it. And it is so rich in nutrients that if I start placing some plant cells over it, they will not even require water. They can just manage by growing on this agar, which is there in the Petri plate or in the beaker. Fine. So, and this undifferentiated mass of plant cells is called as callus. So this is termed as callus. Okay, let me write the correct spelling once. So this is termed as callus, undifferentiated mass of plant cells. Now what I will do, I will take this callus and transfer into a beaker 2. And beaker 2 also again contain some agar. So it also contains some agar, and there are a lot of nutrients present in it. And along with nutrients, there is, let us say, oxygen present in it. So what will happen? The callus will start bearing roots. So we have the roots here coming out from callus. Fine, because there are more oxygens in my medium and very little of cytokinin. So the ratio of oxygen is very high compared to cytokinin. And if it would have been, oh my God, cytokinin is written horribly. Yes. Now, if the ratio had been reversed, so let us say, again, you are taking the small rooted plant so if I am again having a callus, and this time my nutrient medium is, let us say, more rich in cytokinin compared to oxygens. So what will happen? Your callus which has developed roots earlier will now start forming. Why? Because cytokinin is more and oxygen is less. Fine. So you see, it is a concentration of cytokinin and oxygen that determines the formation of root or shoot in a cell culture media. Fine. So what we have done over here is cell culture. We are growing cells of the plant in a tissue culture lab, right? Or in a flask. So this is called as plant tissue culture. Similarly, you can use various nutritive media in agar, which can promote the growth of bacterial cells over it, which can promote the growth of animal cells over it, of different animals and different types, including human cells. So you can literally do tissue culture or cell culture of any type of plants or any type of organisms just by changing the nutrient media. Fine. So by the effect of nutrient media, we have been able to realize that cytokinins promote shoot formation and auxins promote root formation. Okay, let's move forward further and let us talk about the growth inhibitors now. And when I'm talking of growth inhibitors, okay, Nivasini is also having some trouble. So when I am talking of, oh my God, what it is writing? Growth inhibitors. So when I am writing of growth inhibitors, it will include my abscisic acid, also termed as ABA. So it will include my abscisic acid and my ethylene. And the abscisic acid is also called as the stress hormone of the plant. So in animals, the stress hormone was, what was the stress hormone in case of animals? Guys, come on. Let's make it quick. Stress hormone in case of animals. Yes, it is adrenaline. And in case of human beings? In case of human beings, come on. In case of human beings, it is noradrenaline. Noradrenaline. Another name for noradrenaline is your norepinephrine. Another name for adrenaline is epinephrine. Okay, so let us see why we are calling this abscisic acid as a stress hormone. So anyway, I have already talked about auxins and gibberellins. Now let us talk of abscisic acid. And abscisic acid is also termed as stress hormone. Why? So when my plant is facing water shortage, at that point of time, it should close its stomata and should not lose its water through stomatal opening in the form of transpiration. So to close its stomata, it should lose the water from its guard cells. So what tells the plant to lose its water from its guard cells? Abscisic acid. So the abscisic acid released will tell the guard cells to lose out its water and thus become flaccid. So my guard cells, which were swelled up earlier, are now flaccid and my stomatal openings are closed. Fine. And due to this, my plant can bear the stress of less water better. Fine. So that is the role of abscisic acid. Another role of abscisic acid is that it basically promotes abscission. That is why it is called as abscisic acid. The word abscission means cutting. So it promotes tearing off or cutting up of or falling off fruits and leaves from the main tree plant. Fine. So that the plant is having a lot of reserves with it. The plant will quickly suck up the extra nutrients from the leaves that it has to shed, and then the leaves are shed off. Fine. When the abscisic acid is released. So therefore, abscisic acid is helping plants to live in adverse climate during autumn especially. So it promotes abscission, and also it promotes seed dormancy. So if I have a seed and the seed is viable and living, and it contains a living embryo inside it, and it can sprout out. So to prevent the sprouting of the seed under harsh conditions is better, because otherwise the young seedling will die in the harsh conditions. So therefore, during that time, the seed is made to stay dormant by release of abscisic acid from the cells of the seed itself. Fine. And the hormone which works antagonistic to abscisic acid in this regard is gibberellin. So gibberellin tells your seed, wake up from dormancy and start germinating. So gibberellin causes germination of the seed. Okay. Okay, I think what is a dormant seed? We have just talked about it. Now, let us quickly rewind the physiological role of my various plant hormones. So leaf abscission, auxins and ethylene and ABA. So basically, auxin will ensure that a leaf will fall off from a plant only when it is old and it has completed its life. It will allow a fruit to fall off from a plant only when it is old, mature and ripe. Abscisic acid will also help it. Fine. And ethylene is a gas which promotes ripening of the fruits. Fine. So it increases the ripening rate. Apical dominance is promoted by auxins. Right. And cytokinins will work against apical dominance. We have seen this. Another opposite work of auxins and cytokinins. Auxins, basically, we have seen is causing apical dominance. Cytokinin is working against apical dominance. That is one thing. Right. Auxins promote root formation from the stem cutting. And cytokinins promote shoot formation from the stem cutting. Fine. So, one is leading to the formation of roots, that is auxins. And cytokinin is promoting shoot formation. Seed dormancy, abscisic acid or ABA promotes seed dormancy. Gibberellins are working against seed dormancy. Morphogenesis. Basically, morphogenesis simply means the development of various body parts or organs of a plant. So auxins are promoting root formation, right? Formation of this organ. And cytokinins, the shoot. Fine. So that complete is, or the development. Genesis means development. Morpho means body parts. So that completes the development of the body parts of the plant. Okay. And here I guess this will be my last slide of the day. So which plant hormone or hormones will you use if you want to induce shoot formation in a plant tissue culture? I think this is a very easy one. Come on, Alina, Priyanka, Elena, let us make it quick. Christina, Nivasini, Mariam, Tehsil. My God, everyone seems to be sleeping around. Well, In the first case, the shoot formation is induced by cytokinin. We have learned it. Why you guys are taking this much of a time? Induce xylem differentiation or formation of xylem. And xylem is first linked with roots because it has to suck water from the soil. Right? So xylem differentiation occurs in the presence of auxins. Delay senescence. Which hormone delays senescence? It is definitely gibberellin. Right. Inhibit gibberellic acid induced enzyme production. So gibberellic acid gibberellic acid is a kind of gibberellin. And this will lead to production or formation of some enzymes in the seeds of a plant, and due to that, the seeds will germinate. And if I have to prevent the germination of seeds by the formation of enzymes induced by gibberellin, yes, you can write cytokinins as well as gibberellins in the third thing. Both are equally correct. So, if you want to prevent gibberellin-induced enzyme activation leading to germination of seeds, you have to work against it, and the hormone that works against it is abscisic acid. Right. So that will be the correct answer here. Fine. So, okay, let me tell you one more thing. So, auxin is not a name of a single hormone. It is a type of chemical, and some of the auxins include your indole acetic acid, a natural auxin, also called as IAA. We have got indole butyric acid, which is IBA. We have got some artificial auxins also, right? And that is naphthalene acetic acid. That is Na. Fine. So that is naphthalene acetic acid, which is an artificial auxin prepared by humans to induce rooting in stem cuttings and to promote apical dominance and apical growth. Fine. Then gibberellin is also not a single chemical. It is a group of chemicals including gibberellic acid. Fine. So that is another thing. Okay, I guess that is it for today, everyone. So I think we can wind up here. And in our next class, we'll talk more about the responses of plants to various stimuli. Fine. And then we are done with this chapter. Okay, bye-bye, everyone. Bye, Priyanka. Bye, Elena. Bye, Christina. Bye-bye, guys.